Hello, everybody. So, I wanted to take a moment and kind of step back. We're going through a number of these steps through glycolysis and then the pyruvate oxidation, citric acid cycle, electron transport chain, and then oxidative phosphorylation. But as you go through each one, we kind of get buried in the details, and sometimes we forget to kind of go sort of big picture and see what's really happening here. So, of course, this is all taking place inside of the cell, inside of the cytoplasm, and then some of the events are actually taking place inside of the mitochondria itself within these different regions. Remember that the mitochondria is this double membrane organelle, so that's a little bit different than a phospholipid bilayer. It's actually two phospholipid bilayers, meaning a double membrane. So you can think of it as the one membrane being the inside, which is sort of this inner membrane, which has all these little cristae. And then we have this outer membrane, which is kind of what looks more like um, the bean shell that sort of surrounds it. So what we're talking about here, what we've talked about last week, is sort of discussing how glucose goes from being glucose and turning that into pyruvate. And what we're going to talk about this week is everything that happens to this pyruvate molecule once it goes inside of the mitochondria and goes through its various processes that ultimately generate a bunch of these high energy electrons through these high energy electron carriers, which are called NAD and FADH. In their reduced form, they're called NADH and FADH2. Remember that we saw NADH back when we were talking about glycolysis because it's actually one of the products of glycolysis. In fact, we generate 2 NADH per glucose molecule, and these actually get shuttled in and then can take part in some of the more downstream effects. Um, Importantly, the electron transport chain or the electron transport and the proton pumping that actually generates the majority of the ATP. So when they talk about those 36 to 38 ATP that are being generated, that the lion's share is really being generated by these little ATPase pumps, these little protein complexes that are here and are present on the inner membrane of the mitochondria. So let's go ahead and take a closer look at the glucose glycolysis going to pyruvate. Just a little overview just so that it sort of puts us onto first base to figure out what's going to happen to these pyruvate from now on. Okay, so this figure here should look familiar. This is a figure from your book. I believe it's figure 9-7. And here is the process of glycolysis. So you can see here that we start with the glucose molecule, go through all of these steps that we've gone through in detail in the last week, and then we end up with pyruvate here. Now, this happens in all cells. So here, this steps of glycolysis happens in all cells. But what happens to this pyruvate really depends on whether there is oxygen or no oxygen present. In other words, whether you have aerobic conditions or non-aerobic conditions or anaerobic conditions. So we look here at what happens to the pyruvate that comes from glycolysis. And let's start here with anaerobic conditions because this is really all I'm really going to tell you about what happens with this. So anaerobic conditions, when there's no oxygen, this pyruvate, depending on the cell type, can go through a process of fermentation, either to lactate or to ethanol, all right? So ethanol is what we more often than not, we consider this a byproduct of yeast, right? And then we'll reconsider lactate being a byproduct of muscle cells. But both steps are actually doing the same thing. In fact, what they're doing is they are oxidizing this NADH molecule, all right? The purpose of fermentation, even though the brewers would try to say that the purpose of fermentation, or maybe the brewers and the beer drinkers would say that the purpose of fermentation is to generate ethanol. Well, ethanol is really just a byproduct of NADH oxidation, right? So remember that when we were back here in glycolysis, one of these steps, in particular this step here of taking the G3Ps and converting them into this 1,3-biphosphoglycerate, right, required an NAD plus to be reduced to NADH, okay? Now this becomes useful to us if we have oxygen present, but if it doesn't, if we don't have oxygen present, what happens is NADH starts to build up, build up, build up, build up, and then eventually becomes the rate limiting step in actually generating any more here. So if you stop here because you don't have any more NAD plus to reduce, you actually cannot even generate these 1, 2 ATP per glucose molecule. So that's why fermentation is quite important. Not only for the beer drinkers to get their high ABV, stouts and lagers and all those good beers, and that pyruvate to become ethanol, but more importantly to oxidize or remove this high energy electron from this NADH, converting it back to NAD+. Okay. Same thing is happening here. So in muscle cells, right, this NADH is just trying to be recycled, so we get rid of this high energy electron to make NAD+. Now for the rest of this chapter, chapter 10, we're actually going to talk about what happens when there is oxygen present. So you can see here, when there is oxygen present, we actually go through a process of pyruvate oxidation, where we take pyruvate and we turn it into acetyl-CoA. And this is the step called pyruvate oxidation, and as we oxidize pyruvate into acetyl-CoA, we actually go ahead and reduce an NAD plus into NADH. In the process, we also lose a carbon dioxide. Okay. Now, if you're counting carbons, you see here that we start with a 1, 2, 3 carbon pyruvate sugar, and this pyruvate loses one of its carbons to carbon dioxide, and it generates this acetyl-CoA, where we have 1, 2 carbons, this thiol ester group, so this carbon-sulfur carbon connection, connected to this coenzyme A, or what we just referred to as CoA. So we connect these 2 carbons to CoA. All right. So let's see where we're going from here. So of course we start with our pyruvate molecule. Remember, that's coming from glycolysis. We have pyruvate, and we're going down this pathway. So we're going down the aerobic respiration pathway. So take a closer look at that. And now, so what we have here is we have pyruvate, and then we have that first step. So this is pyruvate oxidation. We go from pyruvate to acetyl-CoA. In the process, we generate one of these high energy electrons. So we've transferred NAD plus to, we've transferred an electron to NAD plus. In other words, we've reduced NAD plus to make NADH. In the meantime, we've also lost a carbon dioxide. You can see it's already being formed here, and it just pops off. This carbon dioxide is then lost. And then we go over here to, so this is going to be one of those carbon dioxide molecules that we're breathing out. Okay. So now we go over here to acetyl-CoA. Now acetyl-CoA enters into what's called the citric acid cycle, or sometimes called the Krebs cycle. But here I refer to it as the citric acid cycle, or the CAC. And what happens with the first step of the CAC is that you take this acetyl-CoA and we'll follow it. So we'll follow these 2 red carbons as they go into the process, and these 2 carbons actually get linked up with a molecule called oxaloacetate. So oxaloacetate is already present in the citric acid cycle. It's already gone through a cycle. It's already gone through the circle once. And then now we're going to be adding these 2 carbons to oxaloacetate. So as we add these 2 carbons, we lose the CoA, so this can be recycled then to go out and pick up another pyruvate. Also, a water molecule is added, and then we see that these 2 carbons get added to one of these central carbons here. Okay, so now we have these 2 carbons, we've attached it to oxaloacetate, and this makes the first metabolite of the citric acid cycle, which is called citrate. So this is actually where the name citric acid cycle comes from, is from this metabolite called citrate. Okay. So now this citrate undergoes a transformation, what's called an isocitrate. So this is an isomer of citrate. And if you look closely, we can probably identify what molecule has been moved. So in fact, this hydroxide group has been moved from this central carbon here to this central carbon here. So you see this central carbon used to have a hydroxide. Now the hydroxide has been moved here. Okay. Now remember, as these molecules get moved around, remember that the enzymes that are processing each of these steps are very specific to the molecule that they transform or that they digest or they mutate or they add stuff to. This goes back to that enzyme specificity. Each of these enzymes will bind. So, for example, the enzyme that binds isocitrate will only bind isocitrate, and the enzyme that binds citrate will only bind citrate. So it knows enough or is sensitive enough to be able to bind one and not the other molecule. So here, this molecule is transformed into isocitrate. Not much else happens there. Now we start to get to the good stuff here. So as we take isocitrate and we move to form what's called alpha ketoglutarate, what happens is that we lose our first carbon dioxide. So here you can see these carbon dioxides in gray, or these carbons that are in gray have two oxygens attached to them, right? So they're highly oxidized. These babies are ready to pop right off. So that's indeed what happens here, is that one of these carbons pops off, and in the process of being removed, this carbon dioxide, one of those high energy electrons is actually transferred over to NAD plus to reduce it to NADH, and then this isocitrate is transformed into alpha ketoglutarate. Right? So you see right here was that carbon dioxide group that now became that molecule of carbon dioxide that you're going to breathe out. All right, here we see another grayed out CO2. It looks like this baby's ready to pop as well. So as we go to the next step here, from alpha ketoglutarate to succinyl-CoA, this molecule will then pop off this carbon dioxide, comes off here, and anytime that we lose one of these carbon dioxides, one of these high energy electrons is also transferred to NAD plus to form NADH. So those are the coupled reactions that are happening there. So carbon dioxide pops off, and in the meantime, one of those high energy electrons goes to. And remember, anytime that you have something that's being reduced, you always have something that's being oxidized. So in this case, what we would say is that in the process of taking alpha ketoglutarate, we would oxidize this to form succinyl-CoA. So here we see this CoA sort of shows up again. So this alpha ketoglutarate is actually another molecule that actually binds to this coenzyme A. So that coenzyme A comes in, the same coenzyme A that brings in pyruvate into acetyl-CoA. That same CoA can also bind to alpha ketoglutarate to form succinyl-CoA. And what we have here is very interesting. So this succinyl-CoA, as it forms a bond here, it forms a high energy thioester bond. Now, I'm calling it a high energy thioester bond because as this bond is transformed, it actually generates enough energy to actually take a GDP, similar to an ADP, and convert it into a GTP. GTP you can think of as analogous to ATP. It's another high energy molecule carrier, and that GTP then can later be converted or transferred to an ATP molecule. So you actually are generating one ATP just in transforming this succinyl-CoA with this high energy thioester bond and transforming it into succinate. So as succinyl-CoA turns into succinate, we actually get a first GTP molecule here. Okay, and then again, this CoA, this coenzyme A, then can be recycled and can grab another alpha ketoglutarate, bring it back in, form succinyl-CoA, make another GTP molecule, or of course that coenzyme A can be used up here as well to form another acetyl-CoA from pyruvate. Okay, so as you guys can see here, we're still following these same two carbons that came in all the way from up here from this acetyl-CoA. That has not actually been transformed to any carbon dioxide molecules yet. It actually has to go through this entire cycle one time, two times before these carbons actually start to be separated out as carbon dioxide. So let's see what happens here. So succinate then is transformed, is turned into fumarate, and in the process, a high energy electron from this high energy electron is transferred to a second type of electron carrier called FAD in order to generate FADH2, and that transforms succinate into fumarate. So in succinate, what you have here is you have a single bond between these two carbons, and in this transformation, in the sixth step of citric acid cycle, you actually form a double bond here. So a high energy electron and proton are actually removed from this molecule, boop, and transferred to FADH to actually form a fresh double bond here between these two carbons. You can see that here in this fumarate molecule where you have this double bond here. This double bond is ripe for the picking for a hydroxide group to be added in. So we actually, in the next step, in the seventh step of the citric acid cycle, we actually add a water molecule to add a hydroxide group, shown here, and a hydrogen group here to break up that double bond that was originally here. So now we can take this molecule here, which is now called malate. This malate then has this carbon here, which can then be turned into a ketone. And in the process, we can harness a couple of high energy electrons, transfer them to NAD plus to form NADH, and then we get back to our starting material. So here is, again, oxaloacetate, which can then go through another turn of the citric acid cycle.